Hello and welcome. This is Tika Maya Chetri here to present you my presentation, and I'll be discussing on the topic cancer biology. Uh, I'll be discussing the most important mechanism of cancer and all the most important topics of cancer. To complete all the topics is very difficult. It's a very vast subject. So I'll be cover most important topics of cancer. In the very first, I'll be talking about the introductory part, like what is cancer and its definition. So disease, which is very difficult for us to conquer, and that disease is cancer. So it's a complex disease which occurs when normal cell mutate by mutation. We know we means that means when there is some change in the cells by different means into abnormal cells, because normal cell would normally die after a certain number of population doubling, then they will die. But in this case, abnormal cells, they do not die, they keep on multiplying abnormally, and then they take over the normal tissue, they invade normal tissues, and eventually harm and destroy the host by invading almost all sets of tissue, and that's what we call as cancer. So this disease is characterized by uncontrolled growth and spread of abnormal cells. So cells which are mutate and become abnormal, the cells spread different parts of the body. And then second one is proliferations, rapid reproduction by cell division. That means there is rapid cell division, and then there is no control on the cell division. The third one is metastasis, so spread and transfer of cancerous cells from the region of origin to other areas with directly or indirectly through blood or whatever be the means. So what do we understand by normal versus abnormal cell growth? So let us see what an abnormal cell's growth would be. So in the normal cell, if you see, this is cell division, and then again there are two cells are growing, and if they if there is damage in the normal cell, they would undergo cell death or cell suicide. But in this case, the cancerous cells, there is abnormality in the very first beginning. The cell keep on dividing, they will divide more, they will undergo change in their appearance, they will undergo change in their character shape, but they will still keep on dividing. And there are four mutations: first, second, third, and fourth mutation. At the end of the fourth mutation, there is multiple hits to the cancer. That means when there is error in a single cell, that may be repaired, but when a multiple error happens, cell is not able to repair, and they become cancerous, abnormal, and start growing uncontrollably. So what are the major causes of cancer? Sorry, how did this tumor cell growth happen? So let us look at that. Now, if it is a normal cell, then here, and then normal cell division to fill the lost cells. So there are some cells which are being lost, and then what will happen? Cell division is accelerated in this place, and this place will be occupied by normal cell. Now these cells which are growing over here, they become distinctly different. That means cells which are growing over in a particular area, their physical characteristics become different from their regular cells and which are surrounding them. When this kinds of changes happen in the cell, these kinds of cells can easily break open from the cellular tissue. So as they break open, they invade the blood cells. So from this neighboring blood cells or bloodstream, it will it's being carried to other part of the body. So that's what we call as tumor growth. And next one is invasion and metastasis. So invasion of neighboring cells. So here they start cell growing, become cancerous, they are showing abnormal behavior, and then they are finally moving to the bloodstream. From there where they are carrying to the other part of the body or the new location. So what are the causes of cancer? So obviously first one is mutation. Mutation is one of the major causes of cancer. So what do we understand by mutation? Mutation is a change in the DNA structure of a cell. If the DNA undergo duplication, if undergo deletion, all these kinds of changes can bring about a change in the DNA structure, and hence if the template undergo a change, the protein produced will undergo change, and then the protein produced undergo change, then the protein which controls cell division will undergo change, and the cells, such cells will convert into what we call as cancerous cell. So of course, mutation, in which side mutation, but what will cause mutation? It may be caused by hereditary. Some cancer have heredity input. That means if parents have a cancerous gene that are transferred to their offspring, and then offspring will show chances of having cancer. Next is diet. Sometimes diet contains carcinogenic substance, especially color substance, and or dyes that we use in our food. This can cause cancer. So actually these are the agents which can cause cancer. Then hormones. Sometimes hormone can overexpression cause hormone cause overexpressions of receptor on the surface. So as the receptor are overexpressed, the hormones are excess in the body, then this will then cells will start growing rapidly, dividing and proliferating, that might be converted into cancerous in certain cases. So radiation leads to cancer because radiation cause DNA damage, and this mutation can be carried from one cell to another. So some chemicals can also cause change in DNA structure. They can get interconnect in the DNA. They can mimic particular nitrogenous base. They can integrate in the DNA, so they can cause change in the DNA likewise. So next is virus and bacteria. So some bacteria and viruses may also change by virtue of their integration in the genomic material, by virtue of being integrated close to the promoter element, by virtue of integrating its oncogene very close to some of the active promoters of human body. They might cause cancer under such situation. So what is the etiology of cancer? So what agent which cause cancer? Physical agent include radiation, which comprise ionizing radon gas, irritants, exposures to sunlight, exposures to excess sunlight, and the UV gets rays present in the sunlight may cause melanoma, which is skin disease or skin cancer. Chemical agents like smoking, which contains nitrosamine, aromatic hydrocarbons. They contain like dietary ingredients like drugs, alcohol, and this can cause cancer. The dietary habits like low fat, high fiber food, like processed food in which chemicals are being used, then grilled food, that is also associated with the cancer. So viruses and bacteria, DNA virus, human papilloma virus, this EBV virus, RNA viruses are also associated with the this gastric cancer. So all of these factors lead to a series of mutation in the cell. So what we have seen before, sometimes there are single mutation in the cells. The error correction systems of the cells can take care of the error correction in the DNA, but when this error correction seems to be sleeping in a cell for a very long time, then there is multiple hits in the cells. So this multiple hits in the cells makes cell more prone to cancer. So the epigenetic non-mutations, a change in the DNA, alter gene expression, loss of DNA methylation, keeps oncogene silent, and acetylation of histone. These are some of the changes which do not actually cause mutation in the DNA, but then they can silence certain gene that they may express certain gene, etc. Gene controlling cell growth and differentiation may get altered. So some of the gene which control cell growth and cell differentiation, because they are gene where there is a requirement of control at the gap phase during cell growth, and when the cell differentiation is happening, this gene become abnormal. Of course, it may lead to cancer, and that states two category of gene get affected: oncogene and tumor suppressor gene. Two genes which are responsible for genetics of cancer are basically because of two genes. Either it is oncogene, which has to be expressed for cancer to be developed, or it is tumor suppressor gene, which supposed to be suppress tumor or prevent tumor formation. And if there is loss of function of the tumor suppressor, it can lead to cancer development. Two gene, oncogene and tumor suppression gene, is very, very important for cancer. So this oncogene, they may produce a hormone which encourages cell division by passing the message to the cell nucleus or binding to the certain receptor. Oncogenes could be growth hormone. They can be part of signal transduction pathway, so they can activate pathways. Certainly they may produce growth hormone promoting substances, or they may be involved in transcription, DNA or protein synthesis, and alter transcription. They may increase the transcription, etc. Mutation in the proto-oncogene may produce oncogene. Certain oncogene which are sleeping in a cell called as proto-oncogene. They may be activated by mutation and may turn into oncogene. In dominant gain of function, mutation in change is in the one copy of proto-oncogene is enough to produce cancer. So if there is gain in function, it is usually a dominant kind of mutation. So a single copy of that is enough to produce cancer. Example, rash of oncogene is come from human virus called the Ross sarcoma virus. So next one is tumor suppressor gene. So this code for anti-proliferation signal. They suppress mitosis and cell growth. If there is DNA damage, they will stop the cell growth. They will arrest cell cycle to carry out repair process. Example, p53 activate during hypoxia and UV damage, because these two cause DNA damage. Then mutation may alter a tumor suppressor gene to happen. So the DNA damage accumulates leading to cancer. If certain mutation in germline of tumor suppressor gene, they may be carried to offspring also, like p53 retinoblastoma. These are tumor suppressor gene. They may be inherited like Lee-Frodman syndrome. That's pancreas cancer. Tumor suppressor gene found to be inherited, and it is recessive loss of function mutation. 2 copies of gene must be lost for a person to actually give rise to cancer. So now the methods of treatment. It can be treated by surgery. The removal of tumor, and while the tumor is being removed, the part of area surrounding tumor is also removed. To make sure the tissue surrounding, they will also remove along with the tumor. So the next one is chemotherapy, one of the antineoplastic cytotoxic drugs which is used to kill growth of cells. So the chemotherapy is one of the major. Then we have radiotherapy, high energy ionizing radiations directed to destroy malignant cells. So if you want to destroy the malignant cell, this is only the effect it provides. The tumor suppressor gene are working fine, because the damage will be recognized for a tumor suppressor cell, and tumor suppressor cells then not allowed to form tumor. And the immunotherapy, use of chemical or microbial agent to induce mobilizations of immune defense system. So immune defense system will get mobilized. They will start working toward the antigen which are being expressed by tumor, which are sometimes novel antigen. Angiogenesis inhibitors, because these cells get lot of nutrition, and that's how they survive. So if we can stop the growth of blood vessel around this tumor, then cancer cell can be killed. So an angiogenesis inhibitors, that means substances will do not allow blood vessels to grow, can help in preventing cancer. The hormone blockers of testosterone, estrogens, and prostate and breast cancer can very useful, because this hormone promotes cell growth to large extent. Than gene therapy replacement of mutated gene is in clinical trials. This mutate in tumor suppressor gene and oncogene can be corrected by a corrected copy of gene, either inside the body or outside the body, and they are retransferred back to the body, and that is called gene therapy. So this is the references that I used. Cancer Biology 4th editions by Raymond, and Cancer Biology 2nd edition by Robot. Thank you.